Hi, everyone. A quick intro for the new faces. We're Code Ocean, and our mission is to make science move faster by empowering research teams with easy-to-use computational tools. Our digital lab allows biologists like yourselves to trust your results with full traceability of data and computation, automating best practices, and ensuring reproducible collaboration between teams. We are built on open source, and we continuously research the best tools for our user community and ensure that they run on our platform intuitively, which brings us to today's focus on interactive visualizations. Speaking today is Emily So, and Emily is a bioinformatics scientist at Code Ocean, and she's studying RNA sequencing for her work at the Princess Margaret Cancer Center. Emily, go ahead.
Okay, awesome. Thanks so much, Elena. So like she said, we're going to be talking about interactive visualizations to empower biologists how does it do this? Hold on. Okay. We're going to be talking about interactive visualizations to empower biologists, specifically relating to single-cell RNA-seq. Today's agenda is as follows. We're going to start with some common challenges for today's biologists that you might see currently in your diverse interdisciplinary teams. Then we're going to walk through an example of a common challenge that you would see in a more targeted area, that being single-cell RNA-seq visualization. And then once we cover the challenges you face in that targeted area, we're going to actually do a walkthrough of a solution and how ideally an end-to-end bioinformatic process can look to empower both the computational biologist and the bench scientist. And at the end, we will have a dedicated portion for Q&A, but if you have questions during the webinar, please feel free to put them in the question section. So first, we're going to get started by talking about common challenges for today's biologists and situations you might find yourself in as a bench scientist. This is a common workflow that you might see currently in your team dynamic. Let's say you are working on a project for which an important result is descriptive visualizations. For example, it might be a UMAP plot, a TSNE plot, describing the interactions and behaviors of certain cell populations. The individual generating these results would be the bioinformatician, who is then communicating maybe with data manager, who is passing in clinical data to them, and a clinician who is there to interpret results. And in these quote bubbles is a common challenge you may see. Incessant back and forth talking about different parameters for results, different visualization tactics, and different interests for the results that are presented. And as you can see, these emails can pile up quite a bit. It can be, actually, I ran this by this person and she would like to do this visualization instead. I like this image, but can we change it to be this parameter? And this can be very, very noisy workflow and overall inefficient. And we want to be able to move towards a solution where each individual can empower themselves within their own field of expertise and simultaneously work together as a cohesive unit. And so in addition to these incessant workflows that we are seeing, we are also seeing that outside of workflows, data by itself as an entity is adding to the issue. Before, in those inefficient workflows, that those may have been with trivial, smaller data sets to which the analysis itself isn't that extensive. So if a clinician needed to go into the workflow and execute it themselves to get a desired result, that may have been possible. But now data sets are consistently growing. More specifically, at a single cell resolution in genomics, data sets are growing to be over 1 million single cells. And these can be gigabytes and gigabytes of data. And as data sets continue to grow, this just requires more extensive setup and is resulting in a more complicated and detailed analyses. And that puts more pressure on the computational biologists to make their workflows robust, standardized, and descriptive. And it also puts pressure on the clinician or the biologist as they are being fed the results via that computational biologist who is responsible for keeping the analysis up to par. And there's a continuation of that inefficient and noisy workflow of the back and forth to generate high quality figures or high quality visualizations. Third challenge that you may be facing is actual visualizations and actual results that you're looking at. We talked about workflows in general and communication issues you may face. We talked about input challenges you may be facing as you continue to work with larger and more complicated data. And now we can talk about challenges that we're seeing in the output of a workflow. We have two images here, both of which are a UMAP plot, which is a dimensionality reduction technique, used to visualize results among cell populations. In this case, we are looking at images correlating to the marker of BRCA1 in breast cancer. This might be of interest to both a computational biologist and a bench scientist, as BRCA1 is a known marker in breast cancer, and it might be of interest to see how its expression or activity is dispersed among a data set. But we have two options for a visualization here. We have a visualization that can limit biologist, that being a static image. If I wanted to change it, instead of BRCA1 to ERBB2, or instead of UMAP, I wanted to plot on TSNE, that automatically causes a back and forth that needs to occur. And also an additional large reliance on the computational biologist to make these changes and allow for extensive scientific discoveries. This is a limiting image. But if we look at the image on the bottom, that being a UMAP plot with BRCA1 as a selection, but you can toggle between markers, that's more of an empowering image and an empowering result. And this is available through interaction, obviously. We have an interactive image versus a static image. And by creating these interactive apps, this is addressing the challenge of visualization and empowering biologists. Now, biologists, bench scientists can explore data tactily and make their own discoveries in their own time and without having to rely on the computational biologist and disrupting a team dynamic. So we've talked about challenges that we have faced in our overall workflows, in our input and our output. And while you could see I did have a theme of genomics within my challenges explanation, I think those challenges could be applicable to biologists of any field. If you work in genomics, if you work in radiomics, if you work in multiomics, it could be any situation you are in which there is an interdisciplinary team, those challenges are present. But more specifically, I'm going to start moving towards a targeted example that we see of a common challenge for biologists and a specific solution that is available to address it. And this example is going to be focusing on the field of single-cell RNA-seq, for which visualization is a large component. Just a brief overview for those who might not be familiar. I'm just going to give a brief overview once again of what single-cell RNA-seq is. When you imagine a group of cells, that being in a tissue, a cancerous tumor, we can understand what single-cell RNA-seq is by understanding the information present in that population of cells. Single-cell RNA-seq allows a biologist or a computational biologist to find extremely high dimensional information or describe highly dimensional data and get RNA activity, DNA activity, modification activity of an individual cell at a very high resolution. If you look at this image here that we have of these groups of red cells here, while they may be in the same cell population or may be of the same cell type, that does not mean their behavior is exactly the same. And that's why single-cell RNA-seq is important. We want to be able to profile every individual cell uniquely so that the behavior and the activity of an entire cell population, that here being the red and yellow cells, can be understood more in depth. And when we talk about single-cell RNA-seq, common analysis steps have been pretty standard over the years. There are techniques for dimensionality reduction and individual queries of interest across different cell populations. The two images you see on the right here are pretty common images that you will see in results. You may have what is called a heat map, which is showing the varying expression of genes among certain cells. And this is an even more important result that you will see, that being a UMAP plot. This will show maybe cell communication between populations, cell behavior similarity across populations, the ability to determine which cell population is expressing what certain biomarker. And this can be really important for drug therapy, drug treatment, drug response. These are common images you will see. They've been pretty standard over the years. So like I said, we're going to get into this targeted example. How can we make visualizations that we just saw in single-cell RNA-seq empowering to the biologist? And I'm going to give a brief overview of the data that I'm going to be looking at in our subsequent walkthrough. Let's say we have a data set of single-cell breast cancer cell data. And our interest in our Shiny app, of our interactive app, is to find differences in certain well-known biomarkers, which are always of interest when it comes to cancer research. You always want to get more information about certain biomarkers that lead to metastasis, resistance, so that we can further study them and understand how to treat patients with those biomarkers really active. This data set that I'm using came originally from a paper that was interested in understanding cell populations in triple-negative breast cancer tumors and how different cell populations within a single tumor interact or behave similarly. And this actual original paper was able to categorize certain subgroups associated with drug resistance, onset of metastasis, sensitivity. And this is the kind of visualizations you will very commonly see. Once again, we have a UMAP visualization of a cell population, and we also have heat maps color coding certain variable biomarkers. And this can lead to the discovery of new biomarkers, new treatment methods, so on and so forth. It can have a pretty big clinical impact when you think about it. And so obviously, when we have these kind of data sets here, we need to have accompanying markers of interest. We need to have specific subsets of plots of interest that we want to visualize. And in this case, we are focusing on the biomarkers of breast cancer, the indicators of breast cancer. And we have two options that could be of interest to the biologist. The biologist might want to explore well-known markers of breast cancer, that being ERBB2 or BRCA1, and those might be of knowledge to the computational biologist and might be results they already generate. But something else I want to point out is, what if there is a biomarker that the biologist is really interested in visualizing, but it is not of knowledge to the computational biologist? For example, there are 3 markers that this original paper pointed out, that being KRT18, COL1A1, and CD14. Because these were uniquely found in this paper, maybe the computational biologist doesn't know about these markers. And so we want to make an interactive app that will allow the biologist to explore these unique markers because of a paper they read, without having to do that back and forth. So we've talked about the data set, the rationale, the topic, and now we're going to actually very explicitly talk about the solution that we have. And this is a solution I will be walking through in the subsequent demo. The solution we have is called ShinyCell. It is built off of the Shiny framework, which is written in the R language, which is a framework specifically designed to create interactive dashboards and basically deployable apps. ShinyCell is a Shiny dashboard that was created from the recognition that as data sets and analyses continue to grow, results are becoming less and less easy to explore, and we need to make it accessible and readable to all. So this Shiny app, which is once again called ShinyCell, it was created and designed to visualize common plots you will see in single-cell RNA-seq. That being, once again, we have our UMAPs and our heat maps. And now users can enter ShinyCell to easily visualize already calculated cell-to-cell, gene-to-gene, cell-to-gene comparisons. And this allows you to also select from a variety of parameters, a variety of markers, and allows you to do a lot of tactile data exploration and interactive analyses on your own time without the reliance on other team members. And once again, we're going to be analyzing that breast cancer data set that I just talked about, with the specific interest of finding differences in expression for new and well-known breast cancer biomarkers. If we take a look at that ShinyCell and that Shiny app, you might be wondering, well, if that Shiny dashboard was developed, what did the workflow look like before? And I'm going to give a brief overview of what that would look like, specifically in our platform CodeOcean, because that is where the walkthrough is that I'm going to be going over. And if without the Shiny app, if you wanted to analyze the exact same data set, you would have to do it in an environment like you see on the computer, usually in an active IDE, where you are actively programming in the language R or Python, which obviously requires some predefined programming knowledge. And you would configure the usual setup for bioinformatics analyses, which is also accompanied by the need or ability to interpret inputs and outputs. And you would also need to configure a separate setup for view by bench scientists. The bench scientist may not be able to navigate the code as easily as a computational biologist, making the differences in accessibility already staggering than what we're comparing to in ShinyCell. So obviously we see a big step forward when it comes to not only the ability to process single-cell RNA-seq data, but be able to process single-cell RNA-seq data and then visualize it in an interactive dashboard app. So now that we've kind of talked about the targeted challenge, the targeted situation, and the targeted solution, I'm now going to actually get into the really fun part and go through an end-to-end walkthrough of how a single-cell RNA-seq process can look in ShinyCell. If you have any questions during the walkthrough, please feel free to put them in the question section of the webinar panel. Before I get into the walkthrough of the actual ShinyCell, I'm going to actually explain first what a CodeOcean capsule is before we actually dive into that product. The CodeOcean capsule that we're going to be working in for our ShinyCell example is going to encapsulate all of our code, all of our data, all of our environment, and all of our timeline for the needed ShinyCell deployment. You might be understanding Shiny frameworks, how they work outside on your local machine, how they work on a server, and this might be a little bit of a shift to understand how it's going to work in a compute capsule. A compute capsule in CodeOcean, like I said, is an entity within our cloud computing platform that aims to create a standard, secure, and executable package. So like I said, it is capturing our entire contents of code, data, environment, results, as well as a traceable timeline to be able to track both your activity and the activity of your collaborators. Not only do we have all these results within our CodeOcean capsule, but we have the ability to capture results traceably. We can see past results that were of interest to us as well as our collaborators and monitor them by date and time. Also, CodeOcean capsules offer the ability to be consistent. We offer a function for our CodeOcean capsules called releasing, which allows you to create a snapshot of your Shiny app at a given time. And any capsules that have been released on the CodeOcean platform are guaranteed to run both at the present time of deploying your app and maybe in the future, such as 10 years from now. My current ShinyCell app is created on R4.2. If 10 years from now, I am running it on the current R version is R5.2, my ShinyCell app is unaffected. It will still run on the exact version that I had set when I deployed it, as well as the exact same environment. It is remaining consistent and standing the test of time. I'm going to go over a quick platform explainer of the CodeOcean capsule so that when we actually dive into the ShinyCell product, you're not going to be maybe a little bit overwhelmed of what we actually see here. So I'm just going to give a quick layout before we actually get into it. What you can see are three distinct panels that we have. We first have the file tree system here, which is containing our code, our data, our environment, and our metadata. This allows you to do organization within your capsule, make sure everything is discrete and hidden correctly to the limitations that you need, and you're able to organize your project in an efficient manner. The main panel that we see here is our no code file viewer. This allows you to view interactive results, interactive HTMLs that you may have generated in the past that you want to present to collaborators or make visible to collaborators. And on the right here, our last panel is our timeline. This is what I was talking about before of generating traceable results and traceable activity for both you and your collaborators. Every timeline activity has a collaborator's full name as they are noted on the CodeOcean platform, as well as a date and the time they acted in maybe a cloud workstation or they were doing a computation, and you'll be able to track activity in that way. And now we're just going to talk about how we actually are deploying our Shiny app, because if you saw the previous layout of the CodeOcean capsule, you might be wondering, well, that's not a Shiny app. How do I actually get into Shiny? So our Shiny architecture in CodeOcean specifically employs the function in CodeOcean called the cloud workstation, which is an active IDE that is operating on a cloud Amazon machine. We can operate between two entities. We have our client and we have our app. Our client in this case is the CodeOcean capsule on your computer. It is on the CodeOcean website, but it is all the information that you can see in your compute capsule when you open it up. When we click the Shiny workstation and we enter into the cloud in our Shiny workstation, we have gone from our client to our app. Our app has two components: the server, which is containing all the which is the server, which is containing all the programmatic functions that are required to generate plots, that are required to respond to actions given certain buttons or certain commands. And we have our UI, which is containing all the aesthetic requests and all the aesthetic designs. What buttons are placed where, what sliders are placed where, what functions do you want users to be able to click on. So we have these two components, and these two components are standard no matter what Shiny app you will deploy. You will always see the server.r and the UI.r. And so as we work in our Shiny app, our server.r and our UI.r are dependent on certain responses from our client, that being our compute capsule. That means we need to organize data correctly, we need to organize code correctly, we need to ensure correct environments are installed, correct packages are available. We make our environment and our packages available through the cloud environment system Docker, so you're able to install all your dependencies through that. And by making this request and response loop, we are able to seamlessly operate in our Shiny app in the cloud. And once a computational biologist pre-configures an entire app through Shiny in a compute capsule, they can securely and traceably share that app with their colleagues, who can then, at the click of a button, enter that Shiny app and do the exact same activity the computational biologist was doing. So this is a quick overview of the exact walkthrough we are going to get into today. We have three components. We have our predefined, our pre-explained single-cell RNA-seq breast cancer data, for which we already described the objective, to find differences in biomarkers and their expression in certain cell populations. Now we're actually going to get into the Shiny app in the CodeOcean capsule. I'm going to give you a quick walkthrough or overview of what we exactly are looking at for our Shiny app. At the click of a button, we're going to enter Shiny in the cloud and go ahead and do some visualizations and some interactions in the Shiny cloud workstation. So if you would give me a moment, I'm going to go ahead and open that up. Okay. So as you can see, we are here in our Shiny Cell deployment, in our Shiny Cell compute capsule. We have our three panels, just as I previously laid out in the slides. We have our file tree system here, which has our server.r, our UI.r, and our breast cancer data. We have our file tree viewer here, file viewer here, which is containing a README of the Shiny Cell package, a descriptor of exactly the objective it aims to achieve, as well as additional tutorials that Shiny Cell's authors have provided to get this Shiny Cell app working with your own data of interest. Maybe you're interested in visualizing colorectal cancer, pan cancer. You can very easily make those accommodations. And on the right here, we have a timeline, which is going to capture each user's activity within the app. You can see for the most part, it's been me, and I've been quite active in Shiny. Also within CodeOcean, when you are working on a capsule that has been shared among a team, you have to, only one user can be in the editing seat at the same time. Editing seat means the ability to run computations, work in the cloud, make edits to files, and this is to avoid merge conflicts. I'm going to go ahead and click Start Editing. And now I have full reins to run any computations or any interactive analyses that I would like within my Shiny Cell compute capsule. Now that I've kind of laid out the overview of the compute capsule that we can see here, I'm going to go ahead and enter into our Shiny Cloud workstation. Like I said before, this is kind of going from the client to the server, and this is available through our cloud workstation functionality. Our cloud workstations can be entered really easily just by simply clicking on this Shiny button here, which is the Shiny logo. I'm going to go ahead and give that a click. You'll be met with a loading screen that is showing how your cloud environment and cloud machine is being configured. Once again, our environment is configured via Docker and remains consistent throughout your use of a single compute capsule. As long as no changes are made, I will run the exact same environment on the exact same machine that I set from the very beginning. For the Shiny Cell capsule in particular, you'll be met with a black screen that has bright blue hyperlinks to every directory within our capsule. You can see we have code, data, environment, metadata, and results. This plays a large part into the organization of your capsule. You want to ensure that your two concrete files for Shiny app deployment, that being server.r and UI.r, are in one of these directories. Because actually, the directory that you click on these hyperlinks must contain that server.r and UI.r. That is what the Shiny server is looking for. And so in this case, because I've organized my Shiny cell capsule, I have put server.r and UI.r into the code directory. I'm going to go ahead and give the code directory a click. And within seconds, you can see we have loaded our Shiny cell dashboard. Like I said before, this is pre-calculated, pre-computed, pre-analyzed breast cancer data. And I have multitudes of tabs and multitudes of options for which to explore a Shiny cell analysis. You can see this is the first dashboard that I end up here. We can visualize cell information side by side by gene expression information. For example, here I have the original identities of cell groups that are pre-annotated. So I have malignant cells, T cells, B cells, macrophage and dendritic cells. And I also have the ability to color grade that exact same plot by biomarker expression. So let's give this a look here. You can see BRCA1 for the most part, while a well-known biomarker, there is no really concentrated expression within a single cell group. So maybe I'm interested in doing a more in-depth, a more novel analysis that will bring me some maybe more a little bit interesting results. I can go ahead and click that drop down that we actually saw in our solution for the visualization challenge, the image that will empower the biologist. So I have now a multitude of biomarkers to choose from. Instead of these common breast cancer biomarkers like HER2, BRCA1, and ERBB2, maybe I want to select one of those novel biomarkers from the original paper that I was talking about that I curated this data from. If I go ahead and click COL1A1, you can see the UMAP changes almost instantaneously. And this is maybe a much more interesting, much more discussion-oriented result. I have the fibroblasts almost have an entirely very large color-graded scheme of being high expression. What is COL1A1? What is its role in fibroblasts? Has it been previously documented in other cancers to be indicative within fibroblasts of activity? Maybe I want to change it to KRT18. You can see the expression has almost entirely shifted to malignant cells. Maybe KRT18 has been previously shown to be related to cell death, cell resistance, stuff like that. Because I can go back and forth between these two biomarkers, maybe I actually want to look at them side by side. I can go back up to my dashboard here and click maybe another plot. Let's go to gene co-expression. So here I have a color graded plot on two fronts. I now have a matrix of colors that will show me co-expression. Let's go back and choose those two biomarkers that I just looked at. We're going to go ahead and select KRT18, and we're going to go ahead and select COL1A1. Now you can see we have those two plots practically superimposed on each other. So we have a very high expression of KRT18 in the metastatic cells, and we have a very high expression of COL1A1 in our fibroblast cells. And you can see actually there's a little bit of overlap in this UMAP plot right here. If we go back to that cell information, those are T cells, immune cells. So maybe there is some co-expression that is indicative of an immune pathway that contains both KRT18 and COL1A1. Maybe if I change this to ERBB2, we can see the overlap here. Not as much overlap, we still have that very strong fibroblast expression in the fibroblast expression of COL1A1. There are many different tabs and many different parameters that can be selected within the predefined subset of Shiny cell configuration that already empower the biologist to do a more tactile and independent data exploration. And let's say as well, we as a bench scientist are not necessarily satisfied with the selection of genes that is available in these dropdowns alone. Maybe we're interested in GATA3 or MAPK3. We could possibly go to the heat map option in our Shiny cell dashboard. This will once again offer relative expression of certain biomarkers across cell populations. A little bit of a different visualization, but still one that might be of great interest to some. I'm going to go ahead and change this to a heat map. And I'm going to go ahead and actually type in the biomarkers that I just mentioned that were of interest to me. I'm going to go ahead and do ERBB2 and BRCA1 as well, but I'm going to do MAPK3 and I'm going to do GATA3. So what are some comparisons that we see? We see that ERBB2 and GATA3 are both highly expressed in malignant cells. We see that MAPK3 is highly expressed in an endothelial cell, and we see similar expression levels for both GATA3 and ERBB2 across multiple cell populations. Maybe this is pointing to a new co-expression result. Maybe this is pointing to a similar pathway involvement. This can be a load of scientific advances or scientific discoveries that you can gain from these types of plots. And this is just in generating new gene names that were not previously available. This only further broadens the horizons for the possibility of tactile data exploration and interactive analysis. If there are any questions regarding something you would like to see in the Shiny Cell dashboard or other information of interest, please do put it in the question section. Elena, are there any questions that you maybe want to read out regarding Shiny Cell?
Sure, yeah, let's go ahead and move into question and answer. We had a lot of great questions in. One is to talk a little bit more about how flexible some of these interactive elements are, like the cell types, just asking around clarity, you know, if they're programmatically defined before Shiny Cell is launched, or how to change those.
Yeah, so when it comes to Shiny Cell and the flexibility of being able to change certain parameters, certain cell types, maybe even the data set as a whole, that is all available through the development version of Shiny Cell. That being a certain function that is built into the Shiny Cell package or the Shiny Cell framework that allows you to reconfigure the entire server.r and UI.r files. And that can be available through our actual RStudio Cloud Workstation. You can see we chose to enter the Shiny Cloud Workstation and jump right into the Shiny server. But if you would like to have more of a debugging environment, then we can actually just enter the RStudio Cloud Workstation, which instead of a Shiny server will meet you with an RStudio server and will allow you to do configurations, make new data files, put new input, change the selection of markers even, if you want to still work on single cell breast cancer data, but instead maybe want to work on other versions of the ERBB gene or the other versions of the BRCA gene, you can change those markers as well. The flexibility of parameters is truly endless when it comes to within the bounds of Shiny Cell, and is all available seamlessly through our RStudio server.
Definitely. Also another question kind of in that vein, does Shiny Cell perform differential expression, or can it take a differential expression result as input?
That's a great question. So depending on your capabilities within computational biology, your capabilities within programming, you could absolutely configure a Shiny dashboard to, instead of showing a UMAP plot like this, which was generated via Seurat, which is a very, very popular genomics R package to generate analyses, you could absolutely configure the Shiny Cell app to instead show a plot relating to differential expression. But that will require some configuration on the computational biology front. That will require you to go, once again, go into that RStudio server, interactively debug, interactively run your app in a development version. I would limit the pre-computations that are done within the actual Shiny Cell deployment. I have worked with Shiny apps before that before having to run the app and actually show the dashboard, a series of computations have to be performed, and that can have significant impact on computation time. But overall, if you would like to visualize those types of results, it is absolutely possible depending on your computational biology capabilities.
Definitely. We have another question about kind of reactivity, and a person has a question about if you can link plots to each other. So, you know, linking heat maps together with UMaps, and if you click in one area of one type of plot, does it change the behavior of another type of plot?
Yeah, it's a great question. So when it comes to linking plots, then it's also, once again, possible. Shiny has, with the server.r and UI.r files, it has the capabilities to execute certain actions depending on a user's clicks or a user's mouse movement. And so you definitely have the ability to, upon the clicking on a UMAP plot, let's say instead of toggle plot controls here, the button name was link to TS&E plot, you have the ability to, once clicking on that button, launch another plot, deploy, render another plot, or move tabs, for example. That's all capable within the Shiny framework in general. Specifically within Shiny Cell might require some modifications, but definitely something you can modify and make available through your Shiny Cell cloud workstation.
Yeah, totally agree. Shiny is an amazing package for, the options of reactive elements that you have available to you, and it's really well documented. So certainly something this person should check out. We have another question about how can I share capsules, and maybe could also talk about sharing individual figures with team members.
Yeah, that's a great question. So that question is kind of twofold. Well, first, we're going to talk about how you would actually save an image of interest, and then how you can then share it.
Mm-hmm.
So now we're going to stay in the Shiny workstation for now and talk about how we're going to save one of these results. Maybe I found this KRT18 biomarker, and my collaborators didn't know about it previously, and I said, I really want to share these results with them so that they can look into this biomarker, maybe in vitro or through another wet lab technique, so that we can actually look at its role in breast cancer. I can go ahead and click this button that was pre-configured to capture a result. If I go ahead and click this button, you will see nothing actually occurs in the app, but what that button does is it saves the PNG of this specific UMAP plot to my results folder in my capsule. My results folder in my capsule will then allow me to track the results that were saved during each Shiny session in that traceable timeline that I saw previously. Now that I have captured this result, I can exit this cloud workstation and return back to my capsule to see the results that I saved. So I'm going to go ahead and click that power button and go ahead and click shut down. This basically indicates the server is being terminated and I'm returning back to my client, that being the compute capsule. Now that I am back here, you can see my most recent activity was the following. I finished working in Shiny for 12 minutes and I took what is called a results snapshot. This is my plot of interest. You can save as many results as might be of interest to you. And you can see here, we have our UMAP plot of KRT18. So now we can actually get into the second part of that question, which is sharing a capsule with collaborators and sharing results. Sharing a capsule with collaborators is pretty much analogous or as easy as sharing a Google Doc. If I click this share button right up here, you can see a current list of collaborators on my capsule. I have shared it with my coworker, Elena. I'm going to actually go ahead and remove her to show you how I can add her back as a process. When I have a list of collaborators who are active on my current CodeOcean deployment, I can go ahead and type in their name. So I have Elena. I'm going to click her name again, and I'm going to click add. This will automatically add her user account to my capsule and make my capsule available to her. I can also change around the permissions that I would like to give Elena for my Shiny Cell capsule. In this case, I'm going to give her editor permissions, as editor is the base level permission that makes it available for users to work in the cloud workstation. If I was to make her just viewer, she would not be able to work in the Shiny server in this specific capsule. She would have to create her own copy. So I'm going to go ahead and click save to save Elena as a collaborator on this capsule. And now she has been automatically added and she can view the contents of this Shiny Cell capsule. If instead I just wanted to share the results with Elena, or more importantly, if I saved results of interest that I was worried I might actually lose in the future and I want to be able to find them in a traceable manner, I can do what is called capturing results. This is available when you have a results snapshot from our Shiny session. I'm going to go ahead and click this chevron or this drop down right here, and I'm going to click create new data asset. Data assets in CodeOcean are easily accessible, immutable versions of cloud storage. It's a way to store my figure in a traceable, untouchable manner so that I can access it in the future. I'm going to go ahead and name this Shiny Cell results KRT18 and give it its corresponding name. And this is the KRT18 UMAP color graded plot that I made in Shiny Cell. I'm going to give it the tag Shiny Cell. Tags are similar to hashtags on like Twitter or LinkedIn that you may currently see that allow you to organize your data assets and easily find them by keywords of interest. Once I click and I enter all this important metadata for my results, I'm going to go ahead and click create data asset. Once I do that, you can see this little bar is going back and forth saying capture results. And now I have a CodeOcean data asset containing that plot. I can go ahead and click view. And now it's going to bring up a series of metadata associated with that result contents that I just saved. You can see in here we have just an output, which is containing logs for my shiny cell session. But more importantly, we have that exact KRT18 image that I wanted to save or share. This data set has a unique ID that I can use to index it or access it in the future. And ultimately, if I wanted to share just this result with Elena instead of the entire capsule, and I just wanted to share this result with her by a way of saying, look at this, this was a really interesting result that I saved, I can go ahead and share it with her in the exact same way. I'm going to go ahead and type in Elena again, at her, and now she will be able to view this data set incredibly easily and actually maybe use it in downstream analyses or use it as an image embedding in a markdown file to collect results. And there are a bunch of options on which ways she can work with this image, but it's a great way to maybe batch share or individually share previous results of images that you got without having to scroll through that timeline, depending on how busy it gets, and find the exact image and having them download it to their local computer. Data sets are a really easy way to organize those types of results that you get and share them basically.
Yeah, awesome, Emily. Yeah, I'm really glad we talked about that because to me, that's really the huge thing that CodeOcean adds on to, the accessibility that can be found with Shiny. There's this full reproducible, traceable sharing process, and I really think that that's helpful to our users.
Absolutely.
Okay, so we have another question about how to adapt this to your own analyses. So maybe if you were going to start modifying some of what we've shown today for your own kind of project, what are some of those modifiable elements you would start with?
That's a great question. So for that, we will have to navigate back to our original Shiny Cell capsule. In the Shiny Cell capsule, for which there is also a publicly available version on codeocean.com, I am currently working on the VPC deployment that we offer. Within the Shiny Cell capsule, we have an attached descriptive README. This contains information for running Shiny Cell on CodeOcean, as well as information from the original authors, such as a table of contents and additional tutorials. If we scroll all the way down, you can see that there is a section called Quick Start Guide. This code block here specifically is integral to adapting Shiny Cell and its set dashboard to configure it to your own data. What you will require is a Seurat object as input. This can be a pre-configured Seurat object or one read from an RDS file. And then through a series of functions that have been made available by Shiny Cell, you can run this function called Make Shiny App, which will actually automatically generate corresponding specific data files for you, and it will automatically generate that server.r and UI.r file. Then the layout of your custom Shiny Cell app with your new data will look exactly like the one that I showed today. It may require certain modifications to show biomarkers of interest or even do the capture results, but you will have the exact same UI layout and tailored to the new data that you pass as input. And that's a super easy way to configure it. And once again, it is made available on the public platform that we will share after this webinar if you would like to go and test that out. However, if you want the full capabilities and experience of sharing your Shiny Cell capsule privately, as well as creating those capturing results that I just showed, that requires capabilities only available within our VPC.
Yeah, definitely. Thank you, Emily. And yeah, that actually covered the last question. People were wondering how they can access this. So we'll make sure to include all relevant links for these resources when we share this webinar. Okay, well, great. Emily, great job talking about all these features today. I really feel like we've had a great discussion. Thank you, everyone, for tuning in, and have a great rest of your day. Bye.